Welcome to the 2021 NAE National Meeting. I am John Anderson, President of the National Academy of Engineering and a chemical engineer. I'm sorry that we cannot meet in person as we did last year, but I am confident that we will be safe from COVID-19 next February, and we will be with you in person in California. The theme of our meeting today is engineering and control of infectious disease. And I'm pleased to welcome students from Cabrillo High School in Long Beach, Samueli Academy in Santa Ana, which was co-founded by Susan Samueli, wife of NAE member Henry Samueli, who was the co-founder and CTO of Broadcom. High Tech High in San Diego, a high school founded under the guidance of a former chair of the National Academy of Engineering, Dr. Irwin Jacobs, and a co-founder of Qualcomm, and UC Irvine campus, the students there. Thank you all for being here. A special thanks also to our long-term partnerships with Kenneth Fisher from Cabrillo High School, Nino Polizzi from Samueli Academy, Vanessa Martinez from High Tech High, and Lenora Pena from UC Irvine. I would also like to welcome the NAE members who have joined us today. Thank you for being with us. Our first speaker today is Dr. David Walt, member of the National Academy of Engineering. Dr. Walt is a member of both the National Academy of Engineering and the National Academy of Medicine. He is the Hans-Georg Wyss Professor of Bio-Inspired Engineering at Harvard Medical School. Dr. Walt's laboratory pioneered the development of microwell arrays, which revolutionized the field of genetic analysis and introduced the idea of digital protein detection by developing a high throughput technology performing single molecule analysis. His research is aimed at applying new technologies to address unmet clinical diagnostic needs. Dr. Walt is the co-founder of Illumina Incorporated, a company focused on rapid and inexpensive genetic analysis and eventually DNA sequencing on a chip. And he has also founded several other biotech startups. He is a renowned researcher and scholar, an educator, and an entrepreneur. Today, he's going to be discussing lessons learned from COVID-19 and how to prepare for future pandemics. Dr. Walt.
Thank you, President Anderson, for that very kind introduction. I'd like to welcome my fellow NAE members and particularly the high school students who are joining today. Get this right. We continue to face a significant global health threat. The numbers are staggering, as you can see in the corner of the slide. 100 million cases, over 100 million cases, and 2 million deaths. I updated this slide Monday morning, and I'm saddened by how many more people have been infected since the last time I updated it, and how many more people have died. It's just a true tragedy. The virus is called SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome Coronavirus 2. The 2 distinguishes it from the first SARS threat that struck around 20 years ago, but it was primarily limited to parts of Asia. The disease it causes is called COVID-19. Coronaviruses, which is what SARS-CoV-2 is, have been around a long time and more typically just cause a common cold, but it's thought that this mutated in animals and then was transferred to humans. There are now, as I'm sure many of you are aware, many variants that are circulating that are more easily transmitted. I'll speak to these variants throughout my talk today. In December of 2019, a cluster of patients with pneumonia appeared in Wuhan, China. Within a few weeks, the viral sequence was made available online, and it was published a few weeks later in the New England Journal of Medicine on February 20th, 2020. Now, 20 years ago, the isolation and viral sequencing would have taken at least 6 months, perhaps longer. Now, sequencing is fast, and the rate-limiting step is isolating the causative agent. How is this made possible? The answer is engineers. Bioengineers engineered enzymes that perform the biochemical sequencing steps. Mechanical engineers developed the fluidic system to move small volumes of fluid deliberately and accurately. Optical and electrical engineers provided the detectors that measure the very faint light signals emitted by the sequencing reactions. Material scientists and engineers helped create and integrate the solid-state circuitry that work in aqueous environments. And computational scientists and engineers developed the hardware and informatics to generate the information from the terabytes of data that are generated during a single sequencing run. A true engineering feat. With the sequence in hand, we could start to understand the viral proteins, its infection mechanism. We could begin to design diagnostics tests and start on vaccine development. Sequencing is now used to identify and trace the variants as they begin to appear in various parts of the world. There were 4 areas where we needed technology innovation. And they're shown here: devices, diagnostics, therapeutics, and data analytics, all of which were enabled by engineers. Here you see photos of some of the devices that have been developed and deployed to address problems that arose during the pandemic. The first area that we began to become a real issue when COVID-19 began to appear was personal protective equipment. The infection posed severe risks to healthcare and other frontline workers. This was a really serious issue because we were putting workers at risk, and everyone was concerned that there wouldn't be enough nurses and doctors to take care of all the patients because the nurses and doctors would get infected and the staff at hospitals would get infected. So there was a need for multiple forms of personal protective equipment, abbreviated PPE, that included face masks, face shields, N95 masks, gloves, gowns. The face mask on the left was created to address human factors. This is an engineering issue. It was interesting because patients couldn't see their primary caretakers smile, which left them lonely and depressed. And so engineers came in and designed something that could be functional, but also get over that barrier. One of the most successful short-term approaches for meeting this need was a process called additive manufacturing. Most of you know this more commonly as 3D printing, which turned out to be both useful for rapid prototyping, but also was used to manufacture literally thousands of devices such as face shields and masks. In the middle of this slide, you can see the face shields are produced at thousands per hour with rapid 3D printing, something that wasn't even possible 5 years ago. So really remarkable engineering feat. The full body protection was also essential because we needed to protect people, but also there was a real shortage in gowns. And so these types of personal protective booths simultaneously increased both patient and healthcare worker safety during testing and treatment. It also conserved the personal protective equipment. For example, here in Boston at Mass General Hospital, which is affiliated with Harvard Medical School, it's the largest hospital in Boston, and the use of these sort of booths saved over 98% of the previous gown usage that was used before these booths were designed and developed. Now, there were also many additional devices. I'm not going to go through all of them, but they were in short supply, supplies such as nasal swabs shown on the left here. Ventilators were created by repurposing existing equipment such as anesthesia gas delivery systems. And there were other devices created that addressed such things as the need for human contact. And as you can see on the left here, I'm sorry, on the right, this is a shirt that was developed so that family and healthcare workers could give virtual hugs to patients. These were actuated through air pumps to sort of give patients a little bit of a virtual hug. So what were the lessons that we learned? Well, first of all aspects of engineering were required. And they were driven primarily by a clinical pull, that is a market pull from clinicians. And when the clinician said, here's a problem, please help us figure this out, that's when engineers and researchers got into overdrive and rapidly prototyped and scaled those instruments, those devices. We also found that additive manufacturing really transformed our ability to meet the needs for both the small devices, but also even the medium-sized devices like face shields. But there was a bigger picture lesson. We learned that although the supply chain is global, when countries need those supplies and materials for their own use, the domestic needs are going to take precedent. The US had real challenges with very simple material shortages such as nasal swabs. And I'm sure all of you experienced the pressure to get alcohol wipes and disinfecting wipes. And these shortages pose public health risks. So these supplies need to be stocked well in advance of the need. We can't wait until a pandemic and then start worrying about the supply chain. The second area that we're going to discuss today is diagnostics. There was an incredible ramp up in the capacity and capability in so many areas during the pandemic, not only in devices and diagnostics, but also, as we'll hear about with vaccines as well. But for diagnostics, never before has there been a need for testing at the scale that's required for the pandemic. The need for testing, just to give everybody a sense of how incredibly urgent this was to scale, the entire global lab testing capacity was a third of what was needed to fully address the testing needs of the pandemic. And I'm talking about for all laboratory tests. We're talking about tests for cancer testing, for diabetic testing, for cholesterol tests, all those tests that you hear about that we get routinely. 3 times that is what's needed for the global pandemic for COVID-19. So first we have to discuss, what do we measure? There's 3 molecular classes that we measure here. The first are nucleic acids. That's this little sort of coil in the center of the virus. And this is a picture of the virus, a cross section. That little gold coil, that's the nucleic acid, which in the case of SARS-CoV-2 is RNA. So the genetic material, unlike for humans, which is DNA, in this particular virus, it's RNA. And that's what we measure typically for nucleic acids. The second class are antigens, and the primary thing that we measure is this spike protein that you've heard so much about. That's this pink protein that decorates the outside of the membrane of the virus and gives it its name, sort of a corona. And then there's a third class, which are really not used for diagnostics, as we'll get to in a minute, but but they're antibodies. And these antibodies are molecules that are produced by the host, that is the person who's infected, and they're designed to neutralize the virus by binding to the viral proteins and prevent it from infecting the host cells. And these molecules can be measured to tell whether somebody has been infected previously. So let's spend a few minutes talking about the course of infection. So this slide, I hope it's not too complicated, but it's important to note that the trajectory of each of these molecules is just an average. Every infected person is going to have a different kinetic profile in terms of when these different molecules appear. So at the far left in the gray curve, you see that's the incubation time. I'm sorry, the gray bar right here. That's the incubation time after which the virus has infected cells, is replicating, and is multiplying. This is the pre-symptomatic stage. And in some individuals, symptoms will begin to appear a few days after infection. So at the beginning of that little gray curve right there. Prior to symptom appearance, as indicated by the gray curve, RNA from the virus is detectable. And then in the red curve, you can see that viral proteins that are referred to as antigens begin to appear. Usually within a week after infection, the immune system goes into full gear and various antibodies are produced, which are indicated by the various blue curves. The production of these antibodies typically removes the antigens, which is called neutralization, and leads to a decrease in the virus. And that's when the virus gets cleared from the body. So the blue curve increases, the red curve decreases because the protein is being eliminated by binding to the antibodies. But it's important to note that that gray curve is still detectable. The RNA is still detectable for weeks after the infection has supposedly cleared. And this persistence is likely due to the fact that the virus leaves residual RNA fragments that remain in the host, in the human, after the virus has been destroyed. And unfortunately is often the cause of what are called false positive results, where somebody has been infected, does not have the virus anymore, but they have RNA in their system, and that's detected, and they get a positive test. So how do we sample? Well, there's 4 typical sample types. We're all familiar with the initial one, that's called the nasopharyngeal swab, the NP swab, which are very unpleasant. I've had a few of those. They go deep into your nasal cavity, and people have been looking for alternatives. So the anterior nares swab, the second one from the left, those sample the nasal cavity, but they're much shallower, and they've proven to be equally effective. And then there are saliva, just spitting into a tube, because saliva actually brings up some of the virus in those people who are infected. And then for some of the tests, you can actually use a venipuncture or just a little finger prick of blood, particularly for testing for antibodies. The first 3 samples can contain the virus or molecules that are released by the virus, such as RNA and proteins. But blood does not typically contain the virus. It only contains antibodies. So where do we measure? There's 3 different locations where these tests can be run: central laboratories, and then there's point of care or point of service sites, which includes physicians' offices, urgent care centers, pharmacy clinics. And finally, as we're beginning to see, there are the direct-to-consumer tests, which are intended to be run at home or at people's workplaces or even at school. And I'm sure some of you have actually seen some of these at your schools. And I'll give examples of each of these in the next few slides. So molecular tests are the tests that measure RNA, and these are the most accurate, and they're also typically run at a central laboratory, such as the Broad Institute's COVID testing lab, which is shown here, just across the river from where I'm sitting in Cambridge, Massachusetts. These central labs test for viral RNA, and they use PCR, which amplifies RNA so it can be detected. As you can see, the process requires lots of people, lots of manual handling, although many of the high throughput labs employ liquid handling robots, lots and lots of engineering. There are many logistics required for central lab testing. So let's think about what are the steps that are involved to get a test from a central lab. First, you have to get the sample collected, usually by a trained healthcare worker. Second, when enough samples are collected, because you can't just ship one sample off every time you collect it, then they're transported to the central laboratory. So there's a delay there. Next, the samples are received by the laboratory, labeled, inserted into the system, tested, and then the test is run. And then finally, the results of the test are sent from the lab to the physician who ordered the test, and then the test is reported back to you, the individual that had the test. No wonder it takes so long to get a result. Typically, it'll take a few days. If you're close to one of these central testing labs, it could take 24 to 36 hours. So we need alternatives. And this is really, I think, the next phase of testing for the pandemic. This is the newer category of diagnostic tests, and they just began to enter the market really over the last few weeks. In fact, the one in the middle was just approved last week, and these measure the viral proteins, the antigens. These tests are rapid. They give an answer between 15 and 30 minutes. You can do them at a point of care like a pharmacy or an urgent care clinic, but they also are now available for at-home testing using tests like the ones that are shown here. In general, these sorts of tests are lower accuracy than PCR tests, but they're intended to be used differently. If you suspect or you need to test yourself because you're going to go out or you're going to get on an airplane or you're going to, you want to go visit somebody still with social distancing, you may want to do this on a routine basis, a few days, because what these tests do is they're able to pick up when people are most infectious to others. And so if you test and they come out positive, then you need obviously to quarantine. But they've got a very high accuracy on the ability to detect infectiousness. They're not so great on being able to tell sort of early stages or late stages of infection. These tests are now about $25 a test, but they're expected to be priced around $5 or so when supplies increase and there's additional competition in the market. Many of the sports teams and colleges that you hear about doing testing use these for daily testing to ensure that there's no infection transmission, and they've proven to be incredibly effective. But we're going to need billions of these tests to be available if we're going to be testing frequently, because just in this country, we've got over 300 million people. And so, 100 million tests is not going to do it. And so this is really going to be a supply chain issue for a while. Now, one issue is the increasing prevalence of the SARS-CoV-2 variants that we've heard about. These variants so far still can be detected with existing diagnostic tests. But if there are any additional mutations, these diagnostic tests may not work anymore. I just want to underscore that there's an incredible amount of engineering that goes into designing and building these tests. Imagine they take all the steps performed in a lab and convert it into something that works simply by placing a sample on one of these little devices. There's no pumps to move the fluids, no electronics, no optics. Engineers are amazing. The last type of test are the antibody tests, and these are really used to measure whether somebody has mounted an immune response to the virus. In other words, they determine if somebody's been previously infected and has developed antibodies against the virus. They're also used to determine community prevalence and to determine how strong the antibody response has been by testing the concentration of antibodies in the bloodstream. There's a few other tests that I think are of interest to you. On the left, as I showed you previously, is one of these modern gene sequencers. And this has enabled the scientific community to rapidly sequence the virus. These instruments are widely available and have tremendous throughput capacity and can now literally test 10,000 samples at a time to see whether somebody has the virus. And so these are going to be used as the prevalence in the population decreases, and also they're now being used to identify the new variants and to keep monitoring for those new variants. And I think in the future, these are going to be the workhorse to really look for the new microbial threats for the next pandemic. On the right-hand side, you actually see a wastewater treatment plant, and there's now testing that's going on in those wastewater treatment plants because obviously we all have to eliminate our waste, and in that waste are infectious virus particles. There's the viral RNA that gets eliminated, and that can be tested to actually look at the prevalence in a community. And so these are also a good tool. So what are the lessons learned? Well, we need to have independent and consistent validation by validation laboratories as opposed to the companies that are actually self-validating. We need to keep supplies and we need to maintain a supply chain. With COVID, we got into a situation where the limited supplies caused different states to bid for them, resulting in supplies being available to those who could pay rather than those states where they were really needed. And this point's important because we need to ensure that diagnostic testing is available to all communities, including disadvantaged communities. And this needs to apply not just here in the US, but also globally. There's many countries that just don't have any testing capacity. We need to choose suppliers in advance so that we're ready. We need to have federal coordination because leaving testing to the states just makes it a completely balkanized type of system. And then we need to have an integrated testing plan for using both centralized and distributing testing. Both kinds of testing are essential. Okay, I'm going to spend a few minutes talking about therapeutics, and there's 4 categories that we need to talk about here. First are repurposed drugs. As many of you know, it takes a long time to get a new drug approved. And so for COVID, what we did was we repurposed drugs. What that means is that we identified existing drugs that had already been approved by the FDA and tested whether they could do things that could inhibit the virus or improve people's symptoms. Millions of compounds can be screened with these robotics platforms, and 2 drugs, remdesivir and dexamethasone, are 2 of the drugs that have been found to be useful for severe forms of COVID. They were both discovered using this approach. Convalescent plasma, taking the blood from somebody who's recovered, putting them into somebody who's sick, that's a well-established treatment for infectious diseases. And that was used for a while, but what we ended up doing is developing what are called monoclonal antibodies, which are antibodies that can be directed specifically to some of the viral proteins, and they bind to the viral protein and prevent it from infecting cells. The idea is the same as what was used for convalescent plasma. The antibodies bind to the proteins and prevent it from entering the cells. Eli Lilly just received approval for a therapeutic antibody a few days ago. These are used to bind and eliminate the virus before it has a chance to replicate and get into patient cells and get out of control. But these kinds of therapeutics need to be administered early when symptoms are mild or moderate. Finally, there's vaccines, but before I get to vaccines, it's important to mention, and I think this is really sort of a win for the community, that between the repurposed drugs, the monoclonal antibodies, and changes in patient care that we began to recognize, such as putting people on their stomachs instead of their backs when they're ill, the clinicians have reduced the severity of the disease and the mortality, the deaths from the disease by roughly 50% since the early days. This is still a very dangerous virus, but the chances of survival and the reduction in the disease severity have been significantly improved. So with the variants that are appearing so far, all of these interventions are still effective, but that could change with additional mutations. Okay, just a couple of final comments. I have to get to vaccines. So vaccines have dominated the news, obviously, since the pandemic began. There's a massive global effort to produce vaccines. There's more than 280 vaccine candidates being developed. Only a handful have so far been approved, 2 in the US, 1 is under approval process right now. Typically, it takes 10 years for a vaccine to be developed and approved. The previous record was mumps, which took 4 years. For HIV, we still don't have a vaccine after 30 years. So fortunately, SARS-CoV-2 was an easy target for a vaccine. It took us less than a year to develop multiple vaccines, some of which have been approved, and others, as I mentioned, are still in the pipeline. There are bioprocessing issues that we have. We still have a huge biomanufacturing problem. We need to vaccinate a substantial fraction of the global population. We need billions of doses because many of these vaccines require 2 doses. So with 8 billion people on the planet, 2 times 8 billion is 16 billion doses. So biomanufacturing is really a shortcoming. The amount of vaccine that's required for COVID is 2 to 3 times smaller than the total vaccine production capacity, manufacturing capacity of the world. That includes influenza, mumps, polio, everything. So it's an extraordinary engineering scale-up problem, both for manufacturing and distribution. And then finally, these vaccines are going to need to be updated. Just as the flu every year, we get a different flu shot with an updated vaccine. These COVID vaccines are going to have to be updated as well. And we hope that we're going to get to the point where we don't have to test them every time because we're going to understand them all the way through. So the lessons learned, rapid drug discovery is possible through repurposing, and sophisticated biologicals and vaccines can be rapidly developed, but we need manufacturing capacity. The final topic is data and analytics, and I just want to very briefly go through this. We have mobile health. These are the health apps that you can sort of answer questions about your symptoms on a sort of a daily basis. And these data are being collected to identify early signals from individuals. There's also contact tracing. This is the idea of being able to detect when an infected person has come in contact with other people so that they can be notified to go and quarantine and get tested. These use Bluetooth technologies. So in summary, there's some big picture lessons that we touched on today. There's a tremendous collaboration between clinicians, engineers, scientists, unprecedented development of many new technologies across the spectrum of things that I talked about today. Pandemics are a global problem and they require global solutions. We need to invest in public health systems, both domestically and globally. Engineers have enabled every aspect of the pandemic. One type of engineering that we have not talked about that we probably should add to the list is social and behavioral engineering. And I'm not talking about controlling people, but we need to figure out better ways of communicating the value of these remarkable innovations, inventions, and developments to all those who will benefit from them so we can avoid a repetition of this pandemic. And finally, it's not over. We need to get to where we need to be with diagnostic testing. We need to continue to work on new therapeutics. We need to scale up vaccines and implement mobile health and contact tracing. And also, just as a reminder, until enough people are vaccinated, wear your masks, wash your hands, and socially distance. I've tried to provide some lessons learned today so we can help improve our preparedness for when we experience another pandemic. It's not a matter of time. It's not a matter of if, it's just a matter of time. So we as individuals, our nation, and the entire global community need to be prepared. Thank you for giving me the opportunity to share my thoughts with you today. Happy to answer any questions.
Dr. Wall, thank you so much for an excellent update. This is a subject ripe with questions, and we have received several dozens of them already, and very practical ones indeed. But since we have limited time, I have taken the opportunity to select a few of them that I think could benefit an answer from you. I think much needed guidance here. But let me start off with a very high level question. You have highlighted numerous engineering contributions to sequencing, for example. How was that all initiated and coordinated from the get-go? I mean, in other words, was it a gradual effort or a sudden effort?
Are you talking about with respect to the pandemic?
Yeah, sequencing efforts, yeah.
Yeah, well, that's a great question. Sequencers are pervasive. Almost every country has one, and China, in Wuhan, the folks who first identified patients with this unknown disease that caused pneumonia, they isolated some pleural fluid from those individuals, isolated some nucleic acid, and then just did huge amounts of sequencing on those. And it was really just an extraordinary effort. Within 2 weeks of the first infection, they had the sequence posted publicly. So really, just kudos to the Chinese team that did that. But also, I think it just underscores just the power of being able to do this high throughput rapid sequencing.
Thank you, David. A question from one of our participants here. If you can get more than one type of vaccination, are you better protected? That is, if you get Pfizer first, then the Oxford vaccine as well.
I think, there's a lot of discussion about that in the community. I think at this point, until there are some controlled clinical studies that show the mixing of sort of the prime and then the boost with two different vaccines, I think I'm uncomfortable. I would say if you get the Pfizer vaccine, get your second shot of the Pfizer vaccine, which I'm going to get tomorrow.
Very good. How fast can a mutation occur with this virus here?
How fast
what are you learning? What are you and your colleagues learning?
I'm sorry, how fast can it?
A mutation occur. A mutation occur.
Mutations occur all the time. The real, so we're seeing lots of mutations, and if you, I don't know if it's on the web, but if you go look at the sort of taxonomy of the virus, you can see that there are literally thousands and thousands of variants at this point. Many of them, all cluster, coming from sort of one mutation, and then they branch off from there. I think what people have to realize is that when the virus infects a person, it replicates, makes billions and billions of copies of itself. And the fidelity of replicating the RNA is not so great. And so, if there's billions of those viral particles that are being replicated, each of which have the chance of having a particular mutation, the goal is to, cure that patient, get rid of that virus so it doesn't spread to other people. And that's the danger right now, is that unless we really scale up the vaccination process, if we have all these incubators, that is, people who are infected and sort of brewing additional virus particles, all of which have the potential to be transmitted with another mutation, that's the danger, is that some of these mutations could begin to escape some of our vaccination efforts and obviously our diagnostics efforts as well. Guru, you there?
So, David, the next question is, what is being done about places where rapid screening is required, such as airports?
It's an interesting question. There's a lot of interesting technologies. The Dutch yesterday announced that they're introducing breath tests at some of their portals. Again, these are not fail-proof diagnostic tests. They're just screening tests. I think, frankly, the short-term solution will most likely be these home antigen tests that I described earlier, where when you're in the queue at the airport, you'll run one of these tests, maybe under supervision of somebody, but that will probably pick up all the people who are infectious. And I think that will be a pretty inexpensive 15-minute kind of turnaround. There will obviously be additional technologies that will come out that will probably have involve spectroscopy of people's breath, or something that is capable of looking at some physical parameters of individuals. But all those are early stage kinds of projects right now.
Very good. A little bit of an international orientation now. Could you please speak about what is being done in the developing world and their needs?
Yeah, it's a pretty dire situation. They're challenged with a lack of testing in most of these countries. We here in Boston have mobilized a laboratory that's helping to send them supplies and train them virtually in places like Nairobi and Malawi and Bangladesh. There's a real need just for funding for healthcare worker staffing to do some of these diagnostic tests. And also just the availability of vaccines is much more limited. Now, there are international efforts, WHO and Gavi, which has a vaccine initiative. They along with the developed world's funding mechanisms are trying to acquire vaccine doses that they can divert to those countries, as well as diagnostic testing capacity. But as we're seeing, in many cases, the prevalence of the disease in places like South Africa is out of control. Fortunately, the severity of the disease in that country turns out to be a little less severe. I think it's simply a consequence of the age distribution, a younger population age-wise, but nonetheless, this is sort of gets back to the first question. It's the breeding ground for mutations, and that's why we're going to see mutations coming from those kinds of areas.
We'll now zoom out for two final questions, since we're limited on time. And here comes a question from an aspirant. If I wanted to go into this career, what major would I have to take in college?
So this is a great question. What I would say, and I'm going to back this up, is that you should just follow your passion. Go into, if you love engineering or science, I encourage you to pick that and go into that. And you don't have to focus on infectious disease. And the reason I'm going to back that up is that I never worked on viral infectious diseases previously. My lab works on tuberculosis, but does not work on viruses. We pivoted completely. We learned about the virus as all the new information was coming on. And so I think scientists and engineers, we're all students. We continue to learn. We read, we learn, we absorb new information, we integrate it, and then we incorporate that into our work. And what I would encourage you to do is do what you love to do, whether it's, become a mechanical engineer, an electrical engineer, a chemist, a physicist, whatever it is, do that. And I'm sure that there will be room for contributions from that area when the time comes. And if you are flexible enough and interested enough to pivot to work on those kinds of important problems.
Let's wrap this up with this question, David. We know you have thought about this, but given the experiences that you and colleagues have had over the past year, especially, where do you see medical engineering going in the next 20 years?
I think we're going to see a transformation of medicine in a number of ways. I think it's going to be more precise because we're going to be making a lot more measurements. We're going to be making continuous measurements over time of people. So we're going to be measuring their changes as opposed to just, taking a blood sample and measuring something at that instant in time. We're going to integrate huge amounts of those data, but also other data about the environment, about their physical parameters with wearable sensors, with how they interact with their phone to extract incredible amounts of information from it to be predictive. And I also see that we're going to really benefit from all the advances in genetics, like sequencing, in a lot of the micro mechanical engineering devices to make things tiny that engineers are really great at doing. Nanoengineering and nanotechnology are going to have a big effect. And so things are going to get smaller, they're going to be more interactive, and they're going to be much more information rich.
David, thank you very much for an excellent update and insights. Let's all give a virtual round of applause for Dr. David Wald.
Thank you.
John, over to you.
Thank you, Guru. And thank you, Dr. Walt, for an inspiring and informative talk. I would now like to introduce the Gilbreth Lectures. We're fortunate to have with us two distinguished engineers, Dr. Omar Akbari and Dr. Marissa Jablonski, who participated in an NAE Frontiers of Engineering Symposium and are recognized by the NAE as Armstrong Endowment for the Young Engineers Gilbreth Lectures. The Gilbreth Lectures were established to recognize outstanding early career engineers who are also especially gifted in the presentation of their engineering ideas. Funding for the lectures is provided by an endowment dedicated to inspiring excellent young engineers established by NAE member John Armstrong, who is the retired Chief Technical Officer and Senior Vice President at IBM. We're fortunate to have Dr. Armstrong joining us today for these lectures. Thank you, Dr. Armstrong, for your support of this wonderful program. The Gilbreth Lectures are named in honor of Lillian M. Gilbreth. So who was she? Lillian Moeller, her maiden name, received a bachelor's degree in English literature in 1900 and a master's degree in 1902 from the University of California, Berkeley. She was valedictorian of her class at Berkeley and intended to pursue a PhD at Berkeley, but delayed her plans when she married a fellow engineer, Frank Gilbreth. In order to join her husband in the East, she transferred to Brown University to earn a PhD in psychology. The Gilbreths were a formidable engineering team. They carried out time motion studies, often involving their children, to increase industrial efficiency. When her husband died, Lillian Gilbreth was the widowed mother of 12 children. Until fairly recently, Gilbreth has been best remembered as the subject of the memoir Cheaper by the Dozen, from which 2 movies were made. The book Cheaper by the Dozen was written by 2 of her children. Gilbreth is the acknowledged founder of the field of industrial engineering. She extended her experiments into the home in an effort to find the one best way to perform household tasks. She broke new ground in the area of assistance to the disabled, for example, designing the layout of an ideal kitchen for a person with a heart condition. Lillian Gilbreth was the first woman elected to the National Academy of Engineering. She served as personal consultant to Thomas J. Watson of IBM and to President Herbert Hoover. Lillian Gilbreth was a giant in the field of engineering. I would now like to introduce our first Gilbreth lecturer, Dr. Omar Akbari. Dr. Akbari is an associate professor in the Cell and Developmental Biology section within the Division of Biological Sciences at the University of California, San Diego. His research focuses on the basic genetics and physiology of mosquitoes, with the overarching goal of developing innovative and creative biology-inspired genetic control technologies for reducing the burden of mosquito-borne diseases. Yellow fever, Zika, dengue fever, and malaria together infect hundreds of millions of humans worldwide, killing millions each year. There are currently no effective vaccines for many mosquito-vectored pathogens, and mosquitoes are rapidly evolving resistance to commonly used pesticides. His work develops approaches that can lead to long-lasting solutions. Before joining the faculty at University of California, San Diego, Dr. Akbari was an assistant professor of entomology in the Center for Infectious Disease Vector Research at the University of California, Riverside. He received his bachelor's and master's degrees in biotechnology and a PhD in cell and molecular biology from the University of Nevada, Reno. His presentation is about using CRISPR gene editing methods to combat human disease vector. Dr. Akbari, the microphone is yours.
All right, hello everybody. Before I begin, I just want to thank the National Academies of Engineering for nominating me to be a Gilbert lecturer. I'm really excited to be here today with all of you. I wish I could be there in person to meet you, but unfortunately, given the current pandemic, that's not possible. Today, I'm going to talk to you about using CRISPR to combat human disease vectors. And just going to start off with sort of the impact that mosquitoes have on humanity. Essentially, they're considered to be the world's deadliest animals because they kill more people on Earth than any other animal. It's estimated that about 1 million people die per year from a mosquito pathogen. And if you compare that to death caused by other animals, the next closest thing is humans killing each other. And that's because we love going to war. So why are they so deadly? Well, mosquitoes are so deadly because they transmit so many different pathogens that can affect human health. They transmit malaria, dengue fever, West Nile virus, yellow fever, Zika virus, etc. It's estimated that from malaria alone, we have over 200 million cases per year, and with roughly about 1,000 people dying every single day. And unfortunately, these are mostly children under the age of 5, and it equates to about a child dying every 2 minutes. Dengue fever is also pretty significant. We have more infections than we do with malaria, about 390 million cases per year. A lot less deaths, about 50,000 or so deaths per year. West Nile virus, yellow fever, and Zika are also pretty significant issues. It's estimated that roughly half of the world, 3.2 billion people, are currently at risk of being infected by a mosquito-borne pathogen. Well, how do we protect ourselves from mosquitoes? And really, our first line of defense is to reduce contact. We can do that by using insect repellents, bed nets, lures and traps. We can modify the environment. We understand that mosquitoes lay their eggs in standing water. So if we can prevent the prevalence of breeding sites, we can reduce population numbers in that way. We can use insecticides and larvicides, things that can kill the adult mosquitoes or even the larvae before they mature into an adult. We can use pathogen removal drugs, things like artemisinin, which are used throughout Africa today to combat malaria. And then there's the new approaches, which are the genetic technologies, which I'll talk about today. I wanted to point out that while there are a number of different methodologies used to prevent mosquito bites, none of these are really perfect. They all have issues. Repellents, bed nets, lures and traps, these things cost money. They require continuous application and they need to be distributed to the people that need them. Modifying the environment, you can imagine an environment like so, on the top right, where you have sort of a really wet, arid, humid environment. It would be not practical to modify that type of environment, and it would result in drastic ecological implications if that were to be done. Insecticides and larvicides, these are expensive, they're short-term solutions. Mosquitoes have evolved resistance to many of them, and after the application's over, the populations just bounce right back. Pathogen removal drugs, these are not 100% effective, and there has been documented resistance to these as well. And then many of the genetic technologies that are used today are not sustainable, and they require huge effort. So while all of these approaches are really critical for any kind of sustained success, what we need are new technologies that can be used alongside these approaches. I wanted to point out to you that the mosquito is what we call an obligatory vector for pathogen transmission. And what I mean by that is if a wild mosquito bites an infected individual, the mosquito is then going to take up and ingest the virus or the parasite and incubate it for a period of time, roughly 8 to 10 days. And then it's going to go and feed on another individual and transmit. And this is really the cycle of how these pathogens get transmitted. So how do we block this very simple chain of transmission from mosquito to human, mosquito to human? And really the holy grail is to develop effective vaccines. If we can vaccinate everybody for malaria, dengue, yellow fever, Zika virus, we could potentially solve the problem that way. Well, the truth is that there's been a lot of work in terms of trying to develop effective vaccines for many of the mosquito pathogens, but to date there is not really effective solutions for many of them. An alternative approach is to engineer the mosquitoes themselves to be immune to the disease or pathogen. And the point I want to make here is that both of these approaches can be used to block this simple chain of transmission. So over the last 15, 20 years, there's been some really exciting research in terms of engineering mosquitoes to be unable to transmit pathogens. Here are two of my favorite examples of work that was led by Marcelo Jacobs-Lorena and Tony James, where they engineered Anopheles mosquitoes to be unable to transmit the malaria parasite. There's also been some work in terms of engineering mosquitoes to be unable to transmit the dengue virus. And here's an example of use of RNAi to target one of the serotypes of dengue virus. And this worked quite effectively as well. So when I started my lab back in 2015, it was quite an exciting time. I was starting a new lab, but it was also quite a scary time because it was exactly when the Zika pandemic sort of came to light. And on the news, you would see pictures of like this of babies that had this microcephaly phenotype, and it was very scary. So shortly after starting my lab, I was asked by the university to sort of, if I knew anything about Zika virus, and honestly, I hadn't known anything about Zika virus at the time because it was a relatively new thing. But I quickly became a Zika virus expert, so I started doing a lot of reading about it. I started fielding interviews from the press, and what I learned was that Zika virus was a single-stranded RNA virus, and it encoded about 10 proteins, a few structural proteins and some non-structural proteins, and the genome had been sequenced at the time. And so leveraging some of the prior work I had done with microRNAs, I thought of a strategy that could be used to potentially block the transmission of Zika virus in mosquitoes. So what we did was we actually, taking the sequenced genome of the Zika virus, we designed a set of microRNAs that can target each of these genes pictured here. And we had 8 microRNAs in total, and we drove them the expression of these microRNAs in the mosquito using a promoter that's active in the midgut in response to a blood meal. So when the mosquito takes a blood meal, this promoter turns on and then expresses these microRNAs. Each of these microRNAs, of course, is engineered to target the Zika virus. So if the Zika virus is present in the blood that comes into the mosquito, then these microRNAs can target that RNA genome. And we made transgenic mosquitoes. So on the top right, we have wild type on the left, and then we had our anti-Zika mosquitoes on the right. They have these beautiful glowing eyes. That's how we know they're transgenic. So we engineered these, and then we did this assay here in collaboration with Prasad Pardikar at CSIRO, and basically just challenged our mosquitoes with Zika virus and compared them to wild type to see if they were able to transmit. So we looked at the saliva, which is where they would transmit, and we quantified the level of virus in the saliva, and basically found that our mosquitoes were not able to transmit the Zika virus. So we engineered mosquitoes that were resistant to Zika virus transmission. So, mosquitoes transmit a number of different pathogens, and Zika virus is just one of them, but they also transmit dengue virus. And I mentioned to you earlier that there's quite a lot of people that get infected with dengue virus around the world. So prior work on developing mosquitoes that were unable to vector dengue virus focused only on one serotype. But there's actually 4 serotypes that sort of coexist, and each of them can cause dengue virus. So what we need to do is actually disrupt all the different serotypes that are circulating. And so we designed this approach here, where we essentially use a promoter that's active in the midgut in response to a blood meal. And in this case, we engineered a single chain antibody that was found in humans to basically neutralize all serotypes of dengue virus. So we took this antibody, we engineered it into a single chain antibody, and we expressed it in the mosquito. We made transgenic mosquitoes, and then again, we challenged them with the different serotypes of dengue virus. And we looked at both infection, dissemination, so the ability of the virus to circulate throughout the carcass, and then obviously transmission by looking at the saliva. And essentially what we found was these mosquitoes that we engineered to express this single chain antibody were unable to transmit any one of these dengue serotypes. So this was really exciting work. But what I really want to tell you is that, this observation together with the last couple slides, reveal that we have this power to harness the mosquito immune system and engineer mosquitoes that are resistant to transmitting dengue, Zika, and even malaria. All right, so if we can, if we really want to prevent insect-borne disease or mosquito-borne disease, we need to engineer these mosquitoes to be able to be unable to transmit. And then we need to use them to replace the wild insect population with these engineered counterparts that cannot transmit disease. And the hypothesis is if we have less transmission by mosquitoes, that should equate to less human disease. So this is the way I think about this. This is population replacement, where we replace the wild populations with individuals that are refractory to disease transmission. So the idea is that you have a factory, you engineer your disease-resistant mosquitoes at scale, you release them into a wild disease-transmitting population, your mosquitoes go out, they mate with the wild mosquitoes, they transmit those genes that you encoded in them that can prevent disease transmission. And the goal would be that over the course of several generations, hopefully within a year or two, the entire population would be unable to now transmit pathogens because they encode the genes that you engineered. The grand challenge here is how do we cause a trait or a gene that we engineered in the laboratory to rapidly spread itself throughout a wild population? And the underlying problem here is that these genes that confer pathogen resistance, they likely result in a fitness cost to their carriers. So those mosquitoes that encode those genes, they're not as fit as the wild mosquitoes. They're also raised in a laboratory, and So when you release them into these wild populations, what's going to happen? They're not going to be able to compete. They're going to fall out of the population really quickly, and really nothing will happen. So one solution to that problem is to link those genes for pathogen resistance with what we call a gene drive system. Now, gene drive systems have this notorious ability to be able to spread in a population, even if they decrease the fitness of the animals in which they reside. Now, when CRISPR-Cas9 sort of came into the limelight, there was a lot of prior work done on gene drives, but CRISPR-Cas9 really accelerated the development of gene drives. And developing a gene drive using CRISPR-Cas9 is relatively simple. Really, all one needs to do is encode in the genome the Cas9 gene, a guide RNA that's going to target the gene of interest, and a cargo, which would be your anti-malaria, anti-dengue effector gene, right? You combine all those together, you encode them in the genome, and you position them opposite the guide RNA target site. And when you do that, the cell now encodes the editing machinery. And you drive your Cas9 expression in the germline. So it's active in the germline. And when you do that, what happens is you can get cutting of the opposite chromosome and then repair by homology-directed repair, which essentially copies it over to the opposite chromosome. And when this happens in the germline, and this individual mates out to a wild-type individual, the same process happens in their germline, and you get copying. So what you do is essentially convert what was a heterozygote into a homozygote in the germline. And that process just continues on and on and on until you get fixation in the population. So of course, this technique can be very powerful. It can be used to alter an entire population. And here's a kind of an example of population replacement type of gene drives, where one would release a small number of these individuals into a population, and they would rapidly increase in frequency until the entire population has the gene drive. In principle, a single organism release into a wild population could spread that trait throughout the wild population. And these can be used for driving an engineered anti-pathogen effector gene into a population. And the last point I want to make here is that ecologically, for population modification, the population would still persist, but it would be engineered. There's also another type of drive that is being developed, and these are called population suppression kind of gene drives. And the idea here is that you release a small number of individuals into a population that encode the gene drive. The gene drive then goes and spreads rapidly into the entire population. And then the population reaches a critical point where it rapidly declines and crashes. And if you compare this to a population modification type of gene drive, this type of system ecologically would result in the population declining and crashing out. And there's a number of different ways to engineer this type of system. And one way is to essentially design the gene drive to target a recessive gene that's required for female fertility or female viability. So essentially, when the drive reaches fixation, everybody has a copy of it. All the females would be either sterile or dead, and the population would rapidly decline. So that was basically my last slide on gene drive. And of course, there's a lot of work going on in our lab and other labs to engineer gene drives in mosquitoes and also in other insects. There's a lot of work, there's a lot of discussion on the ethics of gene drive in terms of, can one ever get regulatory approval to use a gene drive? Primarily because these things can be designed to be invasive and spread beyond borders, which makes them difficult to regulate. So I would keep your eye open for developments in gene drive over the next several years. So far, there has not been any field deployment of a gene drive system, but I think in the coming years we potentially will see some field tests of gene drives. And I just wanted to sort of give this last analogy and comparison here. And if you look at the cell phone in the 1960s, it looked like this top left image here. And of course, the cell phone of today, it looks far different. But if you look at how mosquitoes and insects were controlled in the 1960s, it's actually not that much different than how things are done today. And on the bottom right is actually a picture of, this was back in 2015 when the Zika virus, became a big deal. And this was actually in Florida, where they were doing aerial spraying of insecticide sort of, over neighborhoods, right? And so there just really hasn't been enough innovation in terms of, insect control like there has been in other sectors. So I hope over the next several years, we'll start to see more of these engineered insects used in the field. And I'm excited for that future. So I just want to thank you guys for your time. And I wish I could see all of you guys in person and be there in person. Hopefully things will get better in the near future. We'll all get vaccinated and things will get back to normal. I'm really looking forward to that. I just want to thank the people in the lab, that kind of did the work that I described today. So Anna Bushman was responsible for the anti-Zika and anti-dengue, developing those anti-dengue effector mosquitoes. Ming Li, Ting Yang, and Nikolai Kandel, they're working on developing these homing-based drive systems that I sort of mentioned. And of course, some of the grad students, Stephanie Gamis and Michelle Bowie, some of the techs, Junru and Isaiah, and all of our collaborators and funders. And I want to thank the National Academies of Engineering for having me here today. Thank you so much.
Corey, thank you very much for a cutting edge presentation here. We have a number of questions. Let's just get started since we are constrained on time. One question reads as follows: what other species could gene drives be developed in?
Thanks, Guru. That's a great question. Yeah, there are a number of species that people are thinking about in terms of developing gene drives. Mainly focused right now on disease vectors, invasive species. There's been some work in rodents as well. There's excitement in terms of using gene drives to potentially eradicate invasive rodents on islands. In terms of what species it can be engineered in, it's almost limitless. There are some requirements though. The species needs to be sexually reproducing in order to build a gene drive. And also you have to think about the genetics of the species in terms of the development time. So, if one were to develop a gene drive in a species that takes 50 to 100 years to reproduce, then you would only get a doubling every 50 to 100 years. So it could take quite a long time to see any kind of impact. So fast reproducing species are definitely, would work better, right? But I think, the first gene drives that we'll see, tested, if we get there, will be primarily in insects, I think.
We have a number of questions, all about mosquitoes, of course. So once enough engineered mosquitoes are eventually released, how long do you think it'll take for a consistent lowering of infections to a certain degree?
That's a great question. Yeah. So the nice thing about working with mosquitoes is that they have a quick reproduction time. So, they reproduce every 2 weeks. So, and the gene drive is designed to spread pretty rapidly. So it's estimated through mathematical models that depending on the population size, so as population size grows, it takes longer. But if you have a small population size, like let's say an island population, you could get complete fixation of your gene drive within 10 to 15 generations, right? And that equates to about a year to 2 years, right? And you'll start seeing some epidemiological impacts.
The next one, from a design perspective, you can engineer a mosquito to not carry a disease. But what about all those wild ones that aren't engineered? Won't they continue to spread the disease?
That's a great question. Yeah, so the gene drive is designed to spread into the entire population. So even though you'll have mosquitoes that are potentially infected and ones that are not, the gene drive will spread into both types, right? And hopefully hit a point where it reaches fixation. And so everybody will encode the effector gene that can block the pathogen transmission. And so when that happens, you will hopefully will no longer see transmission.
When mosquitoes are artificially mutated, do their offsprings automatically carry all the mutations?
That's a great question. It's a genetic question. I mean, mosquitoes are diploid like us. And so, when a mutation happens and they mate, and they mate out, you get Mendelian transmission, meaning, that mutation, if it's on one chromosome, it's going to pass on to half their progeny and the other half are not going to get it, right? And so, so not all progeny will get it, but, if you link a mutation with a gene drive, right, then you could potentially bias that transmission. And you basically bias Mendelian transmission. You can ensure that every progeny can receive that mutation. There's ways of doing that, but generally that's not, if it mates to a wild type, it's gonna segregate.
This one's an interesting one and has relevance across many other forms of diseases. The questioner asked, what would the impact be if the diseases gained resistance to vaccines as they have done to some other prevention techniques such as insecticides?
That's a great question, and that's something that, it does translate to a lot of things, even David Walt's talk on, coronavirus vaccines that are being used today, right? We're seeing a lot of variants, as he described, and potentially, if we start seeing more variants, then some of the vaccines that are being used right now may not end up working. And so really the only way to counter that would be to design more vaccines, to keep using sequencing technologies to determine what the mutations are that are causing this resistance to the vaccine, and designing new vaccines that can then be used to as a second round or a third round, and you keep going, right? So I think it's true that just like with coronavirus and mosquito viruses also are mutating in the wild, and if you sequence them, you can see that there's tons of variants, and for many of them there isn't great vaccine still, right? So there is a really good vaccine for yellow fever, which is a different type of vaccine than what we're using for coronavirus. It's actually, the yellow fever vaccine is an inactivated virus, and so there's different kinds of vaccines, but that vaccine actually to date, it's been around for a while, and it's a single dose vaccine, and you get lifelong immunity. So we haven't seen, mutations arise in yellow fever that can sort of prevent the utility of that vaccine. But, potentially in the future, we may end up seeing some because viruses are mutating. And when that happens, you have to come at it with another version of the vaccine.
Very good, very good. You had said that you had engineered a mosquito to not have Zika virus. One attendee is asking, how would you be able to do that with every single one?
Right, that's a great question. So yeah, we were able to target the Zika viral genome with microRNAs because the Zika viral genome was sequenced at the time. And we engineered those mosquitoes in a laboratory. So I guess the question is, how would we make all mosquitoes now unable to transmit Zika virus? And really, the way we would do that is by spreading that effector gene into the population in the wild. And the only real way to do that is by using a gene drive system. So we would have to link that effector with a gene drive, get regulatory approval, and public acceptance, and first do a trial and then potentially spread it more widely. But that's how we would use that technology and spread it into the population.
Time for a couple more questions, and then we'll begin to wrap up here. A a general theme of many questions that have come about naturally is, or the kinds of consequences that could ensue because of this work, unintentionally, of course. So should we be concerned about the animal bird populations that will feed on the modified mosquitoes? And a related question is there a danger to use these gene drives on mosquitoes? Can it backfire? So kind of gets into policy, ethics, responsible design, even philosophy. How are you thinking about these kinds of issues, Omar?
That's a really great question. And that's sort of like an ongoing discussion, like with the National Academy of Sciences and many others that are involved in developing gene drives. And, you know, the question is like, what are the risks and how do you assess those risks before going forward, really? And there are risks, right? There definitely are risks. But you have to look at both sides of it, right? So what are the risks of not using the technology, right? In places where people are dying, right? And so you have to weigh the benefits with the risks and then make an informed decision. But you know, there are some risks out there that would definitely need to be assessed before releasing something that you have no control over. So I would say the biggest problem right now with Gene Drive is that it's kind of a unique technology in terms of regulation, right? It's a technology where you release it, but then after you do that, depending on how it's designed, you may not ever have control over it again, and it may just spread invasively kind of everywhere, which may be what you want, but you only want that to happen if nothing unintended happens, right? But if something unintended happens, you want to have control to be able to pull it back. So there's a lot of discussion on, you know, there's definitely ways to engineer it and where you can have control, where you can confine it to locations. And there's, you know, we're doing some of that work and so are some others. But there are other labs that are only designing them to be sort of really invasive. And so is that the right thing to do, especially given that we don't understand, you know, the consequences and the risks or the unintended things that might happen when you release it? You know, those are kind of ongoing discussions right now. And there are a whole number of things that people have come up with that are potential risks, right? And that, you know, I think a thorough risk assessment needs to be done before doing any sort of release, and hopefully, hopefully we can assess for all those different types of things before using them in the field.
Not sure. The final question is from a technology enthusiast or a critic, and they say, when do you think this technology will be deployed, if ever?
That's a good question. I mean, you know, I think if it is going to be deployed, it's going to be somewhere where there's a lot of pathogen transmission. So probably somewhere in Africa where you're getting a lot of malaria transmission, and probably the first use of it will be to sort of suppress populations of malaria disease vectors. But you know, that question actually has kind of pushed our lab into, you know, we've thought a lot about that question, and so we're, you know, I think in some ways we kind of had taken a step backwards. We realized that this technology is very powerful and somewhat scary. So we've actually come up with ways to engineer sort of safer, more confinable technologies that can potentially achieve the same goals. And we're moving those forward first, right? Which also use like CRISPR technology and things like that. So it's a way of testing those technologies in the field to see how well they work. And while we keep innovating and developing and understanding, you know, people's public perception and you know, seeing if it's possible to get regulatory approval to move forward with a Gene Drive. But I think right now, among the labs developing this, I think it's about 5 to 10 years away before we ever see some sort of a field trial. And the question of if ever is definitely a big question, you know, because it's hard to do something irreversible to the environment, right? And to get it's a lot different than, like, you know, coronavirus vaccines, right? You know, they're made. You know, you can decide if you want to take it or not, right? And some people are saying no to it, which is not the right thing to do. I don't think so. I think everybody should take it, but ultimately you have a choice, right? But with a Gene Drive, if you release it, then there's no choice, right? It's out there, and you can't bring it back. So how do you regulate something like that? And that's really an ongoing discussion right now.
Omar, congrats on the Gilbreth lectureship. Thank you. On behalf of NAE, and let's all give Omar Akbari a virtual round of applause. John Anderson.
Thank you.
Okay, thank you, Dr. Akbari, for a wonderful talk and very interesting look at a very important problem. Our second Gilbreth lecturer is Dr. Marissa Jablonski. Dr. Jablonski is the executive director of the Freshwater Collaborative of Wisconsin, based at the University of Wisconsin, Milwaukee. She is an accomplished water engineer, environmental advisor, and plastics reduction expert who has worked in more than 45 countries. Dr. Jablonski lives her life as part of a global community. She is a founding member and mentor to the University of Wisconsin-Milwaukee student chapter of Engineers Without Borders. And she advises on the design and implementation of projects in Guatemala. She also serves as international coordinator of a Guatemalan-led NGO that builds infrastructure with indigenous communities to meet the needs of 5 groups involved in each project. After earning a PhD in environmental engineering, she moved to Washington, D.C. to work in policy as a AAAS Science, Technology, and Policy Fellow. Dr. Jablonski is an advocate for minorities and women in STEM fields and served as a coordinator of NSS Forte program from 2009 to 2015. During that time, she also developed an internationally recognized project that engaged with informal dye industries in rural India to affordably clean their wastewater. Dr. Jablonski's talk is titled Innovative Water Systems, Making the World a Smaller Place. Dr. Jablonski, the microphone is yours.
I want to start by thinking about a couple of the best harmonizers in music history, John Lennon and Paul McCartney. And I know that until your voice has been heard, you are not truly present. So I need you all to sing with me. Please remember, it's very important that your voice leaves your body. It doesn't matter whether you're on or off key. I think we should give this a try. Let's all sing an A. And now anyone who can, harmonize by singing a C sharp. Great job. So now that you've all used your voice, I know you're present. And now that we just harmonized like Paul and John, I know we know about harmony. So let's talk about development engineering. I've been working in the field of development engineering for about 13 years. This is engineering in developing communities all over the world, communities with high need and often limited formal education. And I'm a water engineer. Most of my experience has been in Latin America and Asia. It's worth mentioning that the World Health Organization states that infectious water-related diseases are a major cause of morbidity and mortality worldwide. Although a significant proportion of this immense burden of disease is caused by classical water-related pathogens, such as typhoid and cholera, newly recognized pathogens and new strains of established pathogens are being discovered that present important additional challenges to both the water and public health sectors. Understanding why pathogens emerge or re-emerge is fundamental to effective water resource management, drinking water treatment and delivery, and can have repercussions for countries at all stages of development. I often work with communities in the Ixil region of Guatemala, designing and building water distribution and sanitation systems. And I also work in India with wastewater treatment. I've worked with the United States Agency for International Development, and now I work at the Freshwater Collaborative of Wisconsin. It's my experience that the engineering design implemented is rarely the one originally defined, and that's what I'm here to talk to you about. Engineers are not taught to be communication facilitators. They're hired to solve problems, defined problems. But the job in development engineering includes communicating and complexity. The gap between development engineering and regular engineering is that the problems in development are often not clearly or linearly defined. They're complex problems masked as a simple engineering problem. It might look like the need for a water system, or the need for a bridge, or the need for electricity. But I'm here to tell you that that's just a mask. How many times have I begun a water project to find out that it's not just water that the village needs, it's economic development, gender equality, a voice in policy creation, and much more. In development engineering, the problem solved is not equal to the problem originally defined. People don't often ask for what they want. They ask for what they think their audience wants to hear. When they meet a water engineer, especially from the US, they ask for a water project. This is often misinterpreted as swindling or taking advantage, and it makes most engineers from the US uncomfortable. I call it resourceful. I do the same thing. But this means that in the field of development engineering, design constraints are misinformed. And in the middle of a project, important information can surface regarding priorities, lifestyle, politics, and key players. And we begin to realize that design constraints are often built on assumptions that engineers make and that communities make about each other and about themselves. So I just had you sing a note and then asked some people to harmonize. When you were singing, who were you listening to? The person on your side? Were you listening to yourself? To me? To the person behind you? You were making noise and you were also listening. And I'm here to tell you that you need to listen more. You need to harmonize. Innovative engineering is listening. It's not just listening to the person presenting the problem, however. It's listening to the whole community involved with the project. Back in 2010, I worked on a project in rural parts of India to help silk dyers. You see, what I noticed is that informal dyers were dyeing beautifully colored silk to be woven into saris, but they were dyeing these fibers in high concentration solutions and dumping their dye wastewater directly on the soil, leaving it to seep into their natural waterways. So I went there, assessed the problem, and I created a low-cost wastewater treatment method using photophanton oxidation. Because obviously, it's technology that solves problems. And I hung out in markets and had these big rusty metal boxes manufactured. And I found household chemicals that could be used for the process. And we filtered the final product through sand and carbon, a low-cost wastewater treatment. And it worked. The high concentration dye wastewater decolorized and became reusable. And no one is using it today. Why? Because people are people are people are people. The complexity of the project was realized when the stakeholders were interviewed and the Governmental Pollution Control Board stated that if they could find the informal dyers, they would simply shut down the small dye operations if they continued polluting. Not even a cheap wastewater treatment could solve this problem. What this problem actually needed was systems understanding. There are complex reasons in society that force small-scale dyers to pollute. There are economic, social, health, political, and environmental reasons, to name a handful. For instance, no one will pay higher prices for a dyer's work. This limits dyers' ability to pay for wastewater treatment. Cooperatives exist and protect the variability in silk rearing, but not in chemical dyeing. Corruption exists in the system and creates adjustments that do not allow for easy change. The real long-lasting solution is not technology. It's working with the people in the system. Shortly after this project, I began to learn about systems and was speaking with a systems expert about conversations that can be added to projects to ensure everyone's voice is heard. These are facilitated meetings and conversations with people, because engineering is always about the people, and people are amazing. What the project in India really needed was not only a low-cost wastewater treatment method, but also a method to improve the economics of dyeing, a public awareness campaign to make social change and value natural dyes and the true cost of silk, education in the community about the health impacts of current dyeing techniques and alternatives, a way to address the politics of change and engage with all parties, and soil remediation to save the natural environment for the future. Guess what? People run all of these things. We all work in complex systems and live in complex systems run by people. I also do a lot of work in Guatemala. The work started with Engineers Without Borders in 2007, where I met an amazing local Mason and community organizer named Diego Ramirez Cobo. You won't find Diego published in engineering journals or quoted in books. He's not a professor, and he is one of the wisest men I've ever met. He can build just about anything. He knows by sight what a 30-meter elevation drop looks like, and he can identify an air block in a pipe by the sound that it makes, and then he can fix it with a screwdriver. It is from Diego that I learned the main innovation that I want to convey today, and that is to engage with and listen to the wisdom of the people on the ground. Yes, our engineering skills are helpful. Our funding is critical. Our math is important. Our tools assist. But if we do not engage with communities, studies show that what we create for a community will be short-lived. I've seen water tanks turn into landfills after 6 months. The trick is to build with the community, not for them. During the first project that I was the lead engineer for in Guatemala, there were some changes made on the ground that required change in the pipe design. These changes required that the village would need to carry about 150 extra pipe about a mile up the mountain. After analysis, it occurred to me that it had been preventable. I just missed it. It turns out, had we been more precise with the placement of the brake pressure tanks, we could have kept the pipe design as it had been intended. But we forgot to bring a compass. Such a small little mistake. So I asked Diego to please pull together a community meeting. I needed to explain the situation to the community. So everyone gathered and I explained what happened and how the change required more work on their part. And I apologized for not catching it sooner and explained how sorry I was to create more work for them, for making a mistake. The president of the village walked right over to me in front of everyone and put his hand on my shoulder and said, "Marissa, if the whole system laid only on your shoulders, you would collapse. Somos juntos, we are together." Probably the biggest question I ever get asked about the work that I do is, why do you do this work overseas? Why not work in the United States? And I do question it. And so about once a year, I bring it up to Diego. I turn to him and say, what if instead of spending all that money on flights, bringing groups down here to work together, if we just sent the money down and built more projects, more infrastructure? And he always replies the same way. Marissa, we're making the world a smaller place. It's not only the water system we build that's important. We are connecting 5 different groups and collaborating across boundaries. We are bridging gaps in information and understanding. We teach our students that, in fact, there are 5 groups in every water system we work on in Guatemala. The community, the local association of Masons, in this case it's named ummary, the municipal government, the engineering group, which in this case is Engineers Without Borders, and the funders. We do not come in and bulldoze a community with our engineering ideas and brainstorms. We make the world a smaller place when we actually connect with people. And one of the messages that I want to get across to you today is that it's only when we realize what we get out of development engineering that we can fairly give to it. I developed and teach a development engineering course at the University of Wisconsin in Milwaukee that teaches exactly this. So let's talk about it. What do the 5 stakeholders involved in every engineering project in Guatemala give, and what do they get? Communities give extensive labor, supplies, and cash, and they get infrastructure, or in this case, a water project. The local masons, or ummary, give expertise and skilled labor, and they get paid to work. The municipal government gives supplies like bags of cement and truckloads of sand and gravel, and they get a developed project in one of their communities. The engineers, in this case, Engineers Without Borders, gives all of the design documentation, and they get experience that gets them high-paying jobs after graduation. Donors give funds, and they get to feel great about supporting something, and they also get tax breaks. All of these are valid, good, and helpful. They're important and vital to a project. So the first time we explained this to all 5 stakeholders, we noticed an immediate change. We leveled the power dynamics between the US engineers and the Mayan community. The community asked if what we were saying was true. Seriously, they said, the engineers would go home and use the experience gained in their community to get high-paying jobs. As soon as we confirmed, the community started asking for exactly what they wanted out of the water system. I want my tap there. I need some pipe here. Honest communication emerged. People were no longer saying what they thought their audience wanted to hear. Everyone understood what they were getting and what they were giving. So my development engineering story has led me all over the world and includes a stop at USAID. USAID provides development and disaster assistance to help partner countries on their individual development journey to self-reliance and to respond to natural and human-caused disasters. The United States Agency for International Development is a government funding agency that assists with needed development work all over the world through offices in DC and at missions strategically placed around the globe. I personally spent time in Guatemala, East, West, and Central Africa, and Thailand, along with Washington, DC. I've learned a lot in many domains at USAID, but mostly what I learned is confirmation of what I already know. Engineering is all about the people. And today, I am the executive director of the Freshwater Collaborative of Wisconsin. We are a partnership of Wisconsin's 13 public universities, connecting with industry partners, local communities, policymakers, nonprofit organizations, and advocacy groups. Our mission is to train the next generation of water researchers and problem solvers, and to establish Wisconsin as a global leader in water-related science, technology, and economic growth. In addition to preparing future scientists and water managers, our work benefits everyone who uses and relies on water, from farmers to homeowners, outdoor enthusiasts, and manufacturers. We are powered by our multidisciplinary approach. We provide students the opportunity to customize their degrees and focus on some of the most pressing water-related challenges of our time. These include clean water, climate change, reliable water systems, a robust economy, and technological innovation. The 13 public universities of Wisconsin are already home to strong water programs covering diverse and crucial areas of study, many of which I just listed. To meet the challenges we face as a state, nation, and planet, we need to work together. From our individual, independent strengths, we can create powerful, interdependent, collaborative models by training tomorrow's water professionals with the real world skills employers seek, by strengthening individual UW water programs, by building interdependent networks and fostering innovation, by attracting students not only statewide, but from across the country and beyond, and by creating jobs and expanding Wisconsin's stake in the freshwater economy. Because of all these experiences, I am often asked to sit on committees and be in discussions about program formation because I have so much field experience. And my first question is always the same. What does it look like on the ground? If we support this project, how is the community engaged? What do they want? What have they asked for? How do they help? So ask yourself, as an engineer, what will your work look like on the ground? What are its consequences? Is there a way for you to incorporate community voice in your work, in your innovative engineering? Can you take the time and involve community? Can you do a systems analysis? See which stakeholders are at the table and which should be there. And maybe consider that innovation is actually talking to people and making sure their voices are heard in the project. In 2018, I was brought into a 2-month project in Phuket, Thailand, helping 65 hotels reduce their single-use plastics. Every once in a while, I would get asked if there was a technology that could solve the problem of ocean plastics and beach plastics. Is there a perfect technology that can clean every beach and every ocean of micro and nanoplastics? I had to break some news. Of course, there are innovative technologies, but there is no technology that takes away the power of the human being. We all have to make a daily decision to slow our reliance on single-use plastic, to stop the inflow of plastics into our ecosystems. This is a human decision. And I'm asking you to use people, community organizations, discussions, and to take the time to listen. And just like I tell my students, taking time up front and not jumping into engineering solutions so fast is very important, whether you're overseas or at home. So when you think you're done listening, listen some more, so you can better harmonize when you speak. Thank you.
Dr. Marissa Jablonski. First, thanks for your work in my home country, India. Really appreciate it. And especially on waterborne illnesses, the leading killer here. Since you started your talk with the concept of harmony, let's just start our conversation on that topic. Since you are innately dealing with linguistic differences, value differences across communities. So we're talking about engineering systems approaches that harmonize with the culture that you're ultimately serving. In your engineering mind, professional experience, is there a difference between addressing the needs of a culture versus a client, as seen from a corporate perspective?
Definitely. Thank you so much. And I would also like to thank NAE for inviting me today and for denoting me as a lecturer. I'm honored and privileged to be here. Definitely, there is a difference between a culture and a client. And something that we've realized in the different teams that I'm on, specifically with Engineers Without Borders, is the importance of involving social scientists with expertise in community engagement and facilitation and communication, and not expecting our engineers who have a very specific expertise to know everything about community engagement, and really emphasizing that in a corporate culture, we hope that leadership can also understand this importance. One example that I'm thinking of is we had maybe 3 years ago, one of our graduates got a job in a corporate company, I think somewhere between Milwaukee and Chicago in the Midwest in the United States. And he came to me, he called me up and said, I was just invited to this design meeting in regards to designing a freeway system that, you know, a major thoroughfare. And it involved community. There was a community, you know, built where they wanted to expand the highway system. And it was very interesting. I asked simple questions like, were there any community members at these meetings? How are you advertising that you're having a community engagement meeting? Is it in the newspaper? Is it online? Is it on social media? Is there an Instagram page, right? If you want a well-rounded response from the broader community to be involved in your engineering design, how in fact are you engaging with them? Because if they're not showing up and not giving input, it could be actually the way that the corporation or the company or the group is, let's say, falling short of engaging with them in a strong way. And it's really interesting because I can remember taking an engineering ethics course and discussing this exact thing. The idea that is the client the one to please, or is it the role of the engineer to expand the vision and ask the client, hey, could we get more buy-in if we in fact include more stakeholders than are here? Our work may be more difficult, but the end design will be much more culturally sensitive. And to be honest, this can be in the United States. It's not just abroad or in a developing. That's why I always say developing community. We have developing communities in the United States as well. They're just, we're considered a developed country, but we have developing communities. So worth noting. Thank you. Great question.
Do you find there are generational or cultural differences in trying to implement more inclusive solutions? How do you overcome such resistance?
Oh my goodness, that's a deep one. This is a terrific question. So I want to start by saying that it is young students, young professionals, young people entering in to the field of engineering that are our future. I want to start by saying that. I know there's many students on this call, and I hope you understand your power. And I want to tell you to reach out to me if you ever have any difficulty, because I have definitely run into the old way of doing things, and I have been interpreting, my questioning the system has been interpreted as let's say argumentative or as a conflict at times, when in fact I see it as just expanding the view, right? So if we keep doing the things the way that we've always done them, we will get the same result. And in fact, we want our future to look different than our past. With everything that's going on in the global society around social justice and peace and pandemic, we need to look at things differently. So the best way that I've found to engage with different ages and different cultures is to give respect, start with respect, and open some very general questions. Will this ensure that we engage all people? And point out to young people as well to make sure that they join the table. Historically, in the United States, we have about 13% of our engineers are female, for instance. And so really emphasizing that if we have a table of 10 engineers, that means 1.3 of them are female. So if we are using those engineers to design a city street, as one simple example, are we in fact engaging with a full population of people if we have only 1.3 of them with a differing perspective? Now, that's not to say that those male engineers can't learn those perspectives, definitely. But a very effective way to move forward is to make sure that all are welcome, all belong, and all perspectives are true, so that we can, or valid, let's say, so that we can move forward together rather than have these isolated designs that inevitably leave out a voice. And that can be threatening, Guru. So that's exactly what I'm hearing. And it requires sensitivity. It requires sensitivity and sometimes facilitated conversations. Yes.
Well put. Since we are integrally talking about systems analysis, of course, you cannot avoid the political, organizational, and technical features here. But from your field experiences, what are the real reasons why progress in the water sector has been so slow compared to, let's say, others?
That's a great question. There is, when I was studying wastewater treatment, I learned that if a wastewater treatment engineer from the 1950s in the United States, if that person passed away and we were able to freeze that person and re-enliven them today, that they could walk into the wastewater treatment facility in Milwaukee, Wisconsin, for instance, look around and start working. And I was very distraught. I thought, well, we've at least added some computers, right? And they're smaller and more elaborate. And they said, yes, they would have some sort of learning curve, but not completely. And this was very distraught. And so I started looking into it. And I would say the biggest reason is the infrastructure. So wastewater treatment for a city is gigantic, right? Almost always, I know travel is less now, but if you fly in an airplane, watch the city where you land. If you see these gigantic, they're not buildings, gigantic structures or tanks that are circular, and you usually see 3 of them, those are the sedimentation tanks in wastewater treatment. So you fly over them. Usually they put them near airports because they stink. And think about all of the piping required to bring our toilet, our gray water and black water into those tanks. It is extremely expensive, and space is a large factor in that equation. So I think it comes down to money. We are working with a system that was invested in decades and decades ago, and now we tweak it in these little tiny ways. But if we were in India, for instance, the wastewater treatment method that I was using was much smaller, and it was mobile, and it was about the same cost as one of these larger systems once the system was built, let's say. Right? So then what are the benefits? What are the downsides? At the end of the day, we go with what we trust if we can afford it. I hope that answers the question.
Absolutely. Let's get into the topic of collaboration. You are a demonstrated, very effective collaborator. You also have experiences working in Washington, in the political sphere, and especially since we touched on the topic of working across cultures. When demands differ, when the requirements differ, or the perspectives are variable, and conflicts grow, frustrations grow, how do you motivate, shape the dynamics of the team to get to the intended, desired outcome?
Right. I focus on, I call it translation. So when we're talking about 5 different stakeholders in a project, for instance, those stakeholders might change, but there's always 5 to 10 of them, is my experience, which means there's always 5 to 10 different languages you have to learn, right? So even in the Freshwater Collaborative in Wisconsin, our stakeholders are academics, which speak a language of research and grants and fundraising and student engagement and recruitment, that kind of thing. Then industry, they speak a language of profit and return on investment and quick turnaround. Then we've got nonprofits who are really in for community well-being and often struggle with funding, if we're being honest. Then policymakers, they really speak to their constituents. So whether or not they will get reelected is on the table every day. And then lastly, communities. That's really about public health and human beings, families, children, grandparents, that kind of dynamic. So when we're talking about conflicts and shaking things up, it is very common to hear me enter into a conversation and say, okay, we are all speaking a different language here. And I believe we have the same goal. So if we can center on the same goal, and then we can explicitly describe what that goal will look like on the ground for each of the 5 stakeholders, for instance. And I also call it, we often can agree on the what, and the how will look different for each one of us. And that usually gets people on board. You know, I'm thinking about the last example you just asked about, policymakers. Their reality is very different than people in industry or in academia, for instance. And so their how, if we can explicitly state each, then we can respect each, and we can understand how we will in fact work together. Great question.
Last 2 questions before we wrap up. This is a good one. Do you think any changes should be made to engineering education to better prepare engineers to approach international development projects with a more well-rounded, less technical only understanding of the task?
I do think that there should be changes made to engineering education. And I would like to give a shout out to I know about the engineering education department at Purdue University in Indiana is very strong, and they release many publications, many studies every year around improvements. And there has been great amounts of criticism around international development and engineering education, because the way a typical engineer is taught is to solve other people's problems. And if those people aren't able to define the problem correctly, then it's not the engineer's fault. In fact, I honestly believe that engineers should be taught to zoom out, understand that they are but one voice at the table, that they are an important voice, but so are the social scientists, the communities, the politicians, the greater society. And if we are able in the future to connect engineers with social scientists, I think it will be it'll be a brighter future. I think more people will be pleased. Less people will be complaining about the design of our highway systems, or about the design of our sidewalks, or about any kind of design you could imagine that kind of irritates you. If social scientists are involved, that complaint can disappear, because it will tweak the design, and that's quite lovely, I think.
Very good. Marissa, looking back at your own growth as an engineer, now please try to formulate your response as a piece of advice to a young aspiring engineer. So looking back to your own education and professional growth, was there something that you would have retrospectively taken to have made you a better engineer? So in other words, what can the young participants in our gathering today think progressively, proactively going forward?
Well, I have to say that I really resonated with our first speaker, when he said, follow your passion. So I am not a traditional engineer. My undergraduate degree is in environmental science and Spanish. And then I went back to school for environmental engineering. So I have a master's and a PhD in environmental engineering with a concentration in water. And it was my passion for travel and for human beings that led me to where I am today. So I used to get very, very angry about social justice issues. When I was growing up, there were a lot of articles and in the news about factory. For goodness sake, what do they call that? It's at the tip of my tongue. I'm so sorry. In clothing manufacturing, these manufacturing that are really, really unsafe for human beings, and I loved textiles. I'm a maker, so I knit and I sew, and so I went to this. I went to this art exhibit of a gigantic quilt. I'm still thinking of factory worker, but it's not. It's, oh gosh. Anyway, so I went to this exhibit and the artist said, if you are angry about something, it's because you love something else. And in fact, that very day, this is years ago, I realized that I was angry about so much because I wanted all human beings on earth to have clean water, clean air, clean soil, to grow healthy food and live the life of their dreams. And around that same time is when I had a friend living in Uganda. I went to Uganda to visit her, actually go to her wedding. And I came home and I said, that's what I want to do. I want to help communities have infrastructure to meet their needs. And that's when the learning started about development engineering. And that's when I started my master's and my PhD in environmental engineering and water. But it's really that I followed my passion every step of the way. And so my real advice is make friends. You cannot do this alone. So you need study groups in engineering. You need friends. Today, gosh, this week, I just had a call with someone in the water sector in Wisconsin. It turns out I went to college with her, and she's the director of a grand program up on Lake Superior with estuarine research. And she reached out to me and said, we go to college together. So make friends, follow your passion, take classes that interest you. I remember taking calculus because I loved it. And I can remember taking art classes because I loved it. So yeah, the sky is the limit to all young people. And the last piece of advice is, what kind of 80-year-old do you want to be? Follow that path every day. Wake up and ask yourself, what kind of 80-year-old do I want to be? Go there.
Perfect harmonic ending for the presentations today. As we give you a virtual round of applause, Marissa Jablonski, thank you for your energy and big congrats on the Gilbreth Lectureship. This concludes the presentations. I will now turn it over to Al Romig, Executive Officer of the National Academy of Engineering, for the final portion of today's meeting. Over to you, Al.
Thank you, Guru. Well, thank you, Omar and Marissa, for your very exciting talks and a great compliment to what we heard at the beginning from David Walt. What I'd like to now do is virtually give you your certificates. I'm going to read them so that everyone can hear what they have to say, and then we'll make sure that we get them sent to your address of choice. You can perhaps coordinate with Janet on where you exactly would like it sent. But the first one here for Omar reads, the National Academy of Engineering Armstrong Endowment for Young Engineers Gilbreth Lectures. Omar Akbari is hereby honored as recipient of the Gilbreth Lectureship in recognition of outstanding contributions to the field of biotechnology and presentation of his lecture on combating human disease vectors at the NAE 2021 National Meeting. So congratulations, Omar. Next.
Thank you so much, Al. I didn't know if I can come in to say thank you. I'm really pleased and happy, and I feel lucky to be a Gilbreth lecturer, so thank you so much. Well deserved. As I say, both of you should know the check is in the mail. And finally, but not last, this is the one for Marissa. Once again, it's headed by the National Academy of Engineering Armstrong Endowment for Young Engineers, Gilbreth Lectures. And it says, Marissa Jablonski is hereby honored as recipient of the Gilbreth Lectureship in recognition of outstanding contributions to the field of environmental engineering and presentation of her lecture on innovative water systems at the NAE 2021 National Meeting.
I'll also note that both of these are signed by our board chair. We call it a council, but our board chair, Don Winter, and John Anderson, our president. You met John early when we started the meeting earlier today. Just a few concluding remarks. I hope that you all recognized that we talked about really a lot of interconnections, a lot of complexity, that solving the problems for people in society takes a combination of science, engineering, medicine, all dealt within a cultural tapestry of how you have to deal with the fact that people are people and accept and view things differently from one another. Whether it's a matter of do you want a vaccine or don't you want a vaccine, to whether or not some small remote town in some developing part of the world does or doesn't want to go through some ritual in terms of how they want to get the drinking water or whatever. So those deep interconnections of engineering and the rest of the world and how it's there to solve the world's problems is really, really important. And finally, the last thing I want to make sure that we do is recognize the outstanding NAE team that pulled this together. In particular, Janet Hunsaker, who is the director of our Frontiers of Engineering program, and Michaela Curran, who was our director of membership here at the Academy. The two of them and their teams were the ones that did all the legwork to really pull this together and make it possible. So thank you everyone again. Thank you and congratulations, Omar and Marissa, and we hope you all stay safe and healthy. Bye.